Hello, and thank you for joining tonight's virtual town hall, which will address questions regarding the ACR's COVID-19 vaccine clinical guidance. My name is David Karp, President of the American College of Rheumatology. I am pleased to be moderating this town hall along with my colleague, Dr. Ted Michaels. Tonight's town hall is being recorded so it can be made available on the ACR website later this week. At the time of registration for this town hall, members had the ability to submit questions around the clinical guidance document. Many of those questions will be addressed during this presentation. You are also encouraged to submit questions throughout tonight's presentation using the Q&A box, not the chat box. That's not available to you, the Q&A box. We will answer as many of these questions submitted through the Q&A box as time permits. My co-monitor, Ted Michaels, will be assisting and answering questions throughout the Q&A box in real time. Remember, your microphones will be muted throughout the presentation, so all questions need to be written. Before I introduce our panelists, I would like to provide some background on how we got here. Last fall, as we waited for COVID-19 vaccines to become available, the ACR Practice and Advocacy Task Force issued statements about COVID-19 vaccine research, vaccine availability, and the prioritization of different patient groups during the eventual vaccine rollout, as well as other policy-related topics. These documents have been updated as needed and are available on the ACR website. At the same time, the ACR formed a COVID-19 vaccine clinical guidance task force in anticipation of the first vaccines coming out by the end of the year. The task force was charged with evaluating data and information about new vaccines as soon as they were available and providing guidance to address the needs of RMD patients. The group included experts in rheumatology, infectious disease, and epidemiology. The first two vaccines received FDA emergency use authorizations in mid-December. Data about the efficacy and safety of these two vaccines in clinical trial participants was publicly available at the same time. So the task force went to work. Within a few weeks, data was compiled and reviewed, and drafted guidance was developed and presented to the ACR board, which approved it last week and posted a summary on the ACR website. A full manuscript is presently under rapid peer review at Arthritis and Rheumatology, and the ACR will post it on our website as soon as it is approved. The guidance is considered a living document, recognizing that the landscape is evolving rapidly, so the guidance will be updated as additional data and/or vaccines are available. Next. I would like to recognize the task force volunteers who worked tirelessly throughout the holidays. There are two infectious disease experts highlighted in blue on the slide, and two CDC representatives highlighted in red. Next. Now I'm pleased to introduce our esteemed panelists, all of whom are on the task force. Jeff Curtis from the University of Alabama at Birmingham, who chaired the task force. Ann Bass from the Hospital for Special Surgery in New York. Donald Anthony from the VA Medical Center in Cleveland. Cassie Calabrese from the Cleveland Clinic Foundation. And I want to say that Cassie had sort of a dual role here. She is double boarded in rheumatology and infectious disease, so she was sort of a third ID specialist on the panel. Ruben Arasaratnam from UT Southwestern here in Dallas, and Rebecca Sadoun from Duke University. I will now turn this over to Dr. Curtis, who will provide a brief overview of the recommendations and address some of your questions along with the other panelists. Jeff?
Thank you, David. It was certainly a privilege to be involved and engaged with such a fantastic group that had diverse expertise on such an important topic. The charge to this task force was to develop clinical guidance for rheumatology providers on how best to use the COVID vaccines and to facilitate implementations of vaccine strategies for our patients with rheumatic and musculoskeletal diseases. The target audience was ACR members and rheumatology providers, and while most of this really is going to be generalizable perhaps in some of the principles that were applied, it really has a US focus, and obviously context and healthcare settings are important, and so probably that is the spirit in which it should be interpreted, and other settings, other countries might come to different conclusions based upon resource availability or other decisions that they make. The primary products are intended to be the guidance summary. That's the document that you already have and the one that we're going to be briefly discussing tonight. There's a journal manuscript, as David indicated, that's under review that will be a much more complete set of information and treatment with some of the evidence and the citations provided for each of the guidance statements that were made, and also where the task force may have deferred making specific statements, although I'll make a few brief comments on that as well. I point out that this is a guidance. These are not traditional guidelines. The ACR does do quite rigorous and methodologically sound guidelines across both diseases and rather new for vaccines that would span multiple diseases. This is a guidance that was created in recognition of the fact that there's scant direct evidence, but rapidly evolving science. There's very little direct evidence, of course, for use of COVID vaccines in rheumatic and musculoskeletal disease patients. So the process that was used, although methodologically rigorous and has been used in remote times by the ACR, is not the contemporary process used by guidelines that frankly takes a year or more to do well and typically relies on a great deal of published evidence, which in this case there was very little. It's also not meant to replace clinical judgment and shared decision-making with the patient. In other words, these guidance statements are intended to be relevant for most patients in most clinical settings, and in guideline parlance, that would be considered provisional or conditional recommendations. And none of this is intended to supersede the judgment of an individual clinician and what he or she might talk with individuals who might make different decisions depending on their own circumstances. There's also a number of other ACR efforts, some of which are listed, and the goal is to align with and to be concordant with, but not to duplicate many of those other important things that the ACR is doing related to practice and advocacy in this space. Next slide. As part of the charge of what the task force was asked to do, there were a variety of scoping questions that were developed. Essentially, how broad are these guidance statements going to be? Which vaccines should they cover? Should it be all of them in any phase of development? Or should they be only the available ones, or maybe the near-term horizon ones that are in development? Those would be examples of scoping questions. You know, what would be out of scope that won't be covered? So that was agreed upon by the work group members of the task force, as well as the larger ACR vaccine guideline task force, of which this is a part. There were 3 teams so as to facilitate parallel processing. A literature scan was done through January of 2021. One of them was focused on vaccine efficacy and safety for the mRNA vaccines, both of them that are currently available, as well as the 3 additional ones that were felt to be late-stage development and might soon be available in 2021. The second was focused on COVID epidemiology, particularly in what's known in rheumatic and musculoskeletal disease patients, as well as vaccine effectiveness. And here we had the great advantage of the many guidelines that had been developed in times past, and particularly those that had information and a systematic literature review specific to vaccination in rheumatoid arthritis and psoriatic arthritis. So all those literature searches that were done for those disease-specific guidelines that had vaccine information were culled, and all of those original articles were pulled. And then finally, the last was that same corpus of evidence where the focus was safety concerns related to vaccination, such as disease flare or worsening. All of that information was collated into a 70-page evidence summary developed in about 6 weeks. We iterated on that multiple times as new evidence accrued over the course of this very rapid process and distributed to team members prior to rating. Next slide. The process used was the RAND UCLA appropriateness method, which uses a modified iterative Delphi process. We used a 2-round process. It was asynchronous that had electronic voting. The voting, or the rating, if you will, was provided by each task force member, and ultimately across all of the task force members, for all of the rating statements that were achieved with consensus, the consensus was then classified as either being strong or moderate consensus, depending upon how much agreement and the strength of that agreement present across the entire task force. There was a series of 2 webinars to discuss those ratings and to discuss particularly those statements where uncertainty existed, and either email asynchronous or real-time synchronous modifications to some of the guidance statements were made to refine some of the wording in a way that would be most appropriate and germane in light of the evidence and the way that the task force members rated that evidence. Next slide. So I'm going to show you some excerpts and not read all of them. People have access to this. It's on the ACR's website. I've bolded a few of them, of those that I want to make particular mention of. So the third here, based upon their risk for COVID, this subgroup of patients who have autoimmune and inflammatory rheumatic diseases, so that's the acronym that's there, they should be prioritized for vaccination before the non-prioritized general population of similar age and sex. So some of the nuances there were to recognize that patients that are on immunomodulatory drugs that may be somewhat immunosuppressed probably have a higher risk for serious COVID or hospitalized COVID and worse outcomes. But does that mean that, you know, a 40 or 50-year-old woman with lupus is going to do worse than an 80-year-old nursing home resident? Probably not. And that's the caveat about the similar age and sex. Two down, there was also an expectation that the response to COVID vaccination for many patients, not all of them, but many of them, is likely to be blunted in magnitude and duration compared to the general population. We certainly have that expectation for many other vaccines, but it's fairly nuanced depending upon which immunosuppressive or which immunomodulatory drug that's being used. So again, it's a general principle. It's not true of everyone. And of course, there's no direct evidence. And I would be quick to acknowledge that. And probably the task force members, myself included, will remind people of that it's quite clear that we don't have direct evidence. All of this evidence is extrapolated based upon immunologic principles from other vaccines and vaccine platforms, and those principles were applied to what we might expect for COVID vaccination. And then finally, a theoretical risk exists for flare or disease worsening following COVID vaccination. However, the benefits of vaccination for rheumatology and musculoskeletal disease patients were felt to outweigh the potential risk for new onset autoimmunity. Those risks for flare or disease worsening have been observed for other vaccines, mostly at the case report level. There certainly was a case of new onset rheumatoid arthritis that was deemed related and serious in the Moderna program as an example. But the task force also recognized the fact that there certainly have been case reports of new onset rheumatoid arthritis and vasculitis following COVID infection itself. So this is by no means unique to this vaccine or vaccine platform, and in fact has been observed even following natural infection. So it's not even specific to vaccination. Next slide. Probably the most important statement of any of them in the entire document is this first one. Rheumatic and musculoskeletal disease and autoimmune and inflammatory disease patients should get vaccinated, consistent with the age restriction of the emergency use authorization and or perhaps in the future FDA approval. So practically what that really means is that patients 16 or 18 and above should get vaccinated, hard stop. That might seem rather self-evident, and yet there have been other countries, including our neighbors to the north, that at least in some provinces and for a time said, well, there's no direct evidence, and so, you know, patients with rheumatologic or immunosuppressive conditions or treatments shouldn't get vaccinated. So if you don't say something that seems perhaps self-evident to some, you can have unintended consequences where at-risk people are denied vaccination, as we have examples for. The second statement I would also call out as being important, and that is that patients who might be on an immunomodulatory therapy should be vaccinated in a similar fashion, even if they don't have an autoimmune disease. So for example, someone with erosive osteoarthritis that might be on off-label methotrexate, or someone who's on methotrexate, again off-label, who might be on it to treat immunogenicity due to pegloticase, that person would still have the methotrexate specific guidance applied to them, even though they don't have an underlying autoimmune disease. The third statement, based upon what we know at this time for the two mRNA vaccines, there was no preference for one over the other, and patients should receive either vaccine that's available to them. Whatever they do get, though, for the first dose, of course, they should get that same vaccine for the second dose. A somewhat controversial statement that I believe Dr. Bass will speak to a little bit later on this evening. There was not felt to be sufficient evidence for healthcare providers to routinely order any lab testing, such as antibody tests to the spike or nucleocapsid proteins, to either assess immunity to COVID post-vaccination or to assess the need for vaccinated in someone who perhaps had COVID but had not yet been vaccinated. So we'll return back to that, though, in several minutes. And then finally, while vaccination would ideally occur in the setting of well-controlled disease, life is not always so perfect. So unless someone has sick unto death, life-threatening illness, like they're in the intensive care unit, COVID vaccination should occur as soon as possible for everyone for whom it's being recommended, although there are a few treatment-specific considerations that I'll speak to in just a moment. Next slide. So some of the most important topics that I think people were quite eager to have some understanding of what the task force had recommended are shown on this slide. So we've lumped two things together here that I would just point out in this middle column. These are timing considerations for vaccination, as well as the timing considerations for what to do with immunomodulatory therapies around the time of vaccination. So just recognize those are two different things. For the most part, and for all the therapies on those first two very large rows, there was no modification recommended to either immunomodulatory therapy nor vaccination timing. So the patient who's on any of those would get vaccinated as soon as it's available, and you wouldn't do anything to hold or modify or otherwise affect the timing of those treatments. The three exceptions on this slide are methotrexate, Janus kinase inhibitors or JAK inhibitors, as well as abatacept. So for methotrexate, there's a recommendation to hold methotrexate for a week after each vaccine dose. For the two mRNA vaccines we have, of course, that would have you holding for a total of 2 weeks, one after each of the two doses. Similar guidance for Janus kinase inhibitors to hold each of those therapies. And of course, there's 3 currently approved in our field at this time for a week after each of the 2 vaccine doses. And then finally for abatacept, to hold once weekly sub-Q abatacept. And this one's a little bit different. 1 week before and after the first COVID vaccine dose only, and to not interrupt around the time of the second vaccine dose. And we'll speak in just a few minutes to the immunologic rationale for why that was felt to perhaps be different and why this guidance statement is not the same as for methotrexate and JAK inhibitors. Next slide. And then the last set of guidances regarding a few other therapies, so abatacept and cyclophosphamide IV, where it might be a little bit more difficult to interrupt or to hold, there was a set of guidances about timing vaccine administration. People can obviously read those statements and hopefully they are clear, but we can certainly take questions. The one I wanted to specifically call out, given our limited time, is for rituximab. So there's a couple important caveats. The first is that assuming the patient's COVID risk is low or can be mitigated by preventive health measures, you would schedule vaccination so that the series is initiated about 4 weeks before the next cycle. So for example, for someone who's on a 6-month schedule, you would give the vaccine at approximately month 5. If you're giving rituximab cycles every 4 months, you would give the vaccine at approximately month 3. And then if after vaccination, you would delay the next rituximab for 2 to 4 weeks. That if, or the assuming part, that again is assuming that COVID risk is low or able to be mitigated. And on the next slide, I'll speak to one overriding principle that's relevant for rituximab. Next slide. I would point out again in this third bolded statement, there's no direct evidence about mRNA COVID vaccination safety nor efficacy in rheumatic and musculoskeletal disease patients. Nevertheless, on balance, the task force felt that the benefits were going to outweigh the potential risks associated with vaccination. The concerns or the guidances about the timing were predicated not on safety concerns, but really the intent to optimize vaccine response. So that next bolded statement. So the risk of deferring vaccination or delaying vaccination, and thus if you don't vaccinate, you haven't done anything to mitigate COVID risk, should be weighed against a possible blunted response to the vaccine. So the idea was, should you give the vaccine now, immediately, under maybe less than ideal circumstances? For example, someone who's on cyclophosphamide and 1 milligram per kilogram of glucocorticoids. As long as you thought you were going to get some vaccine response, perhaps that's better than not vaccinating, getting no response, and then thinking a few weeks or months down the road, well, perhaps you could measure something or give a booster dose. There's a lot of unknowns about that we didn't have answers for. And in light of evidence about how to delay or test something down the road, defer vaccination, give a booster, a lot of other things that might make sense that you could do without really any evidence or strategies to support what to do, I think on balance, the task force said it's probably better in almost all circumstances to vaccinate more quickly and not to delay and not to defer, because the sense was probably patients are going to get some benefit in the short term and it's not going to be zero. And that's where most of these guidance statements where there was a large gray area that the task force voted. And then finally, that very last statement, there is recognition of some of the principles that I suggested, that you likely could and would be able to give a booster, or perhaps even at some point repeat the series. Maybe some lab-based testing might help us be smarter about how to do that. We don't have a lot of those answers yet, but that's why, on balance, there was the general recognition that probably it's better to vaccinate earlier rather than later, except for a few unique circumstances. Next slide. So to summarize, this is very much the abbreviated version. There is a full manuscript. The manuscript's been written. It's under peer review in arthritis and rheumatology. It's expected to be published in March 2021. It's been optimized by the journal in light of the importance of this. As David already mentioned, this is a living document, so we absolutely expect revisions. It was certainly developed with imperfect evidence and, frankly, a complete paucity of direct evidence. We are in the process of making updates quite regularly based upon new information for the available vaccines. The new vaccines not available in the US as of January 2021, and would include those that are coming before the FDA VRBPAC even in the next 2 weeks. And then finally, we are badly needing COVID vaccine data and the direct evidence that currently we're lacking in rheumatic and musculoskeletal disease populations. So with that, I'm going to turn things back to David and to Ted to field many of the questions that came in from the pre-registration, as well as ad hoc questions that people are submitting throughout the evening. Thank you.
Thank you, Jeff. This is certainly an enormous amount of work to have accomplished in only 8 weeks. We've had a number of questions from people as they registered for the town hall. So let's discuss some of them, and then Dr. Michaels will moderate questions that we've been getting online. And I think that some of the questions we've had already will answer some of our online questions as well. So next slide. So some of the more common questions that have been submitted ahead of time ask for some clarification on the use of concomitant anti-rheumatic medications. Dr. Bass, this was a frequent one. Can you please discuss the rationale and prior data for holding methotrexate and JAK inhibitors for 1 week after vaccine doses, and for methotrexate, why hold 1 and not 2 weeks?
Sure. This is a question that we expected to get. And the reason for that is that Park and colleagues have done a few prospective studies showing that holding methotrexate for 2 doses following the flu vaccination increases immunogenicity. And so why not here as well? And I think there really are 2 reasons. The first is that mRNA vaccines are different. And you can tell they're different in terms of the speed of response. And you can tell that really from the clinical trials where clinical protection from COVID started as early as day 12 after the first dose. So the method of inducing immunity seems to happen more rapidly. So that was one reason to hold for 1 week rather than 2. And then the second is that this is a 2-dose vaccine, and we were concerned that if you held for 2 weeks after both doses, patients would be missing 4 weeks of their medication and that they would be at risk for flare. So those are the major reasons.
Great, thank you very much. Can we have the next question? So, Dr. Anthony, one of the other drugs we got a lot of questions about was abatacept. So can you explain the immunologic basis of holding abatacept post-vaccine and the data supporting this recommendation? Then there was a second question that we wanted to tack onto that. Why weren't belimumab and other medications such as mycophenolate or cyclophosphamide recommended to be delayed post-vaccination?
Yeah, this is a good question, and we discussed this issue. I think that the abatacept, keeping in mind the target of abatacept is the interaction between CD80 or CD86 and CD28. This is the second signal needed for naive T-cell activation. Most of the vaccines that we administer on a routine basis, such as influenza, primarily are reactivating memory cells. In this case, we're intending to activate a naive T cell for the COVID vaccine, and we felt particularly sensitive about this and wanted to direct efforts to window the immune suppressant around the first exposure to the neoantigen during the first dose of the vaccine. I think that there are no data, as we said, that really are supporting this. This is the biologic target, and we felt we'd like to give this recommendation. The other agents listed were thought to be more in the category of agents that were not optional to window because of disease severity or disease activity, or additionally lack of information about immune suppression in regards to other vaccine responses. So we opted to, for simplicity, because of lack of data in these cases, to recommend for the time being to continue the medication right through the time frame of vaccine.
Great, thanks. Now, next slide. Dr. Calabrese, this is one of our most thorny questions. It's one that my patients and other doctors are really having a hard time with. Can you review the data for the timing of vaccination and rituximab? We have a lot of people wondering what to do with the different schedules, like 3 months, every 3 months, every 4 months. But typically, I think the task force addressed the every 6 month schedule. Can you go over that?
Yes, thank you. This is very thorny. As you said, this is a really important question and important discussion point. And we know that of all the targeted therapies we use, rituximab has been shown to have the most significant impact on vaccine responses. And we know this from data on influenza and pneumococcal vaccines. And we know that B-cell depletion and decreased humoral response can be present for up to 6 months after a dose of rituximab. And some thought and consideration to timing vaccines around rituximab is important, and this is supported by ACR and UR guidance in non-COVID vaccine situations. So we have recommended to administer COVID-19 vaccine 4 weeks before a next dose of rituximab is due. So if someone's on a Q6 month cycle, that would be 5 months after their last dose, essentially waiting as long as possible and then leaving a 2 to 4 week window before next rituximab dose is due. If someone's on a Q4 month cycle of rituximab, that would be waiting 3 months after their last dose and 1 month before the next dose was due. And certainly this highlights kind of the overarching principle that our document is guidance and not the law, and our autoimmune and inflammatory disease patients are very heterogeneous, and we'll do the best we can with this. It will certainly be challenging, but it won't be possible to time this perfectly in all of our patients, especially thick vasculitis patients with end organ disease, but these are our recommendations, and that is the reason for them.
Great. Let me ask a sort of a follow-up. Did the task force think about measuring B-cell numbers prior to vaccination, given this data?
That's a great question, and not something that we discussed or chose to recommend, but certainly something to consider and to look at moving forward.
Great. Go to the next question. These next two questions refer to vaccine safety. So, Dr. Arasaratnam, can you speak to the first one? What do you think is the risk of activation of autoimmune conditions or other immune manifestations after a vaccination, or even after getting COVID-19 itself? That's sort of a broad topic, so maybe we just limit to vaccination tonight.
Yeah, well, good evening, everyone. It's great to be here. I think, whenever there is a antigenic trigger, if it is either via infection or via vaccine, there's always the concern of off-target autoimmune effects. I think it's very important that obviously we may have had anecdotal experiences of autoimmune conditions in our patients, and the question is, what is the denominator and what is the actual risk? I think the first point to understand is we shouldn't extrapolate from infection to vaccination. In the setting of an infection and flares, that can be confounded by changes in medications, and also that risk represents an immune response to the whole virus, whereas the currently authorized vaccines actually generate a very focused response to spike protein. So with that in mind, what is really the risk in vaccination? Because I think this is important for patients. As alluded to earlier, in these phase 3 trials that lead to authorization of these mRNA vaccines, there's very careful systematic monitoring of adverse events. And in the supplementary appendix of the Moderna phase 3 trial, there was one case of rheumatoid arthritis in the vaccine group compared to 0 in the placebo, but that's 1 in over 15,000 people who got the vaccine. And so the risk is really very, very low. So what happens when you move from a phase 3 trial to real world effectiveness, and how is that being monitored? Well, I would really encourage your patients to sign up to the CDC vSafe app, responsibly report any adverse effects to the VAERS, the VAERS reporting system with the CDC. And actually, this monitoring of adverse effects is actually a very critical part of safety monitoring that's actually ongoing now and an area of research study. So, for example, there is a network called the Vaccine Safety Data Link, VSD, that has several integrated healthcare systems that's actually monitoring on a real-time cycle about adverse events and potential safety signals with the vaccines. And published on the slides of the CDC website back in January 27th are just a report of just over 100,000 patients who have received the vaccine. And actually, there's no concerning safety signals, particularly also when you look at autoimmune events, so myopericarditis, myelitis, GBS, Bell's palsy. And so what I would emphasize is so far, the risk appears to be very, very, very low. And the overwhelming benefit of vaccinating in this population to prevent COVID-19 is far, far greater than the theoretical uncertainties that we have at this very moment.
Great, thanks. That's a great answer. Next, another safety question that's very common. Dr. Sedin, can you answer this? Can pregnant and nursing women receive the vaccination?
Sure. The short answer is yes. Full stop. The longer answer is that the CDC, as well as the Advisory Committee on Immunization Practices, as well as the American College of Obstetrics and Gynecology, have all come out with statements suggesting that pregnant and breastfeeding women should be offered the COVID-19 vaccines, the mRNA vaccines that are currently available, if and when they would otherwise be offered the vaccine. So now we have to acknowledge that the vaccine has not been around for 9 months, and so the data in human pregnancies is somewhat limited. But one, the data from animal models are encouraging. 2, of the 13 women in the Moderna trial who were pregnant, the only miscarriage occurred in a woman who had received the placebo. Probably most importantly, number 3, is the fact that there are these large national registries right now that are actively collecting information on the many thousands of pregnant women who have received the COVID-19 vaccine, and there have not been any worrisome signals. When you add to that the benefit of women in their 3rd trimester in particular, there is reason to believe that the vaccination will result in antibody transfer and passive immunity following birth. And then finally, there isn't any theoretical basis for concern related to the mRNA impacting the genetics of the fetus, and there is no theoretical basis for concern of any negative impact on milk and lactating women.
Thank you. I've been watching, and there have been a number of great questions that have come up in the question and answer box. So I'm going to turn it over to Dr. Michaels and ask a couple of these questions.
Yeah, thanks, David. Lots of questions, hard to keep up with, actually. So I know a few people have raised their hands, which I know is helpful in a lot of Zoom meetings, but if you could put questions in the Q&A box, that would help tremendously and let us track those and hopefully get answers out to folks. There's a lot of interest in the question, the Q&A box, around the methotrexate issue and around, not surprisingly, the JAK inhibitor issue. And Dr. Bass, maybe I'll throw this at you. One of the questions was sort of timing of vaccination, thinking, pre-vaccination in terms of methotrexate dosing. If the methotrexate is scheduled to be dosed maybe the day before vaccine or after. And then some comment maybe about the week duration of holding the JAK. There was a couple questions around that. I was hoping maybe you could take a shot at that.
Sure. Just to reiterate, none of this is evidence-based, and hopefully down the road we will have evidence to support the recommendations, and maybe the recommendations will change. So These are really guidance to help you think about how to manage your patients. And so with regard to the methotrexate question, I think we were all surprised by the PARC study, where it turned out that holding methotrexate before vaccination really didn't help, but it was only holding it afterwards that boosted immunogenicity of the flu vaccine. However, I agree, if your patient is scheduled to receive methotrexate the day before their vaccination, I don't want to move away from what the guidelines say, but I personally would probably say that kind of counts as the week. But for simplicity's sake, we did say the week after the vaccine, but I think you can use your judgment. JAK inhibitors are, and I should say that the reason that we were concerned about the use of JAK inhibitors around the vaccine was because of the importance of interferon responses to vaccine responses, and interferon signaling being blocked by JAK inhibitors. JAK inhibitors are very fast on and off. So I think that stopping them, the day of vaccination, it's really going to be out of your system quickly, and that timing should be adequate. But I think even looking at the methotrexate study by PARC, these are relative values. Immunogenicity was not as good as it was with methotrexate being continued. And because it had been subject to a prospective study, we felt that this was an area where there actually was some data, which is why we singled methotrexate out. But I don't think any of us think that there would be no response to a COVID vaccine if your patient inadvertently takes their methotrexate, the day after their vaccination.
Thanks, Dr. Bass. I think that was very helpful. There are also some questions. I'm going to turn to our infectious diseases expert here. There were some questions around specific to vaccines and maybe some of the emerging choices that we'll have with vaccines in the hopefully very near future. So there's a question around the adenovirus vector vaccines, assuming those are available within a short time, in terms of considering those as live viral vaccines, as that's been an issue with other live attenuated vaccines in the past. How should rheumatologists and patients think about that vaccine moving forward?
Thank you very much. Well, firstly, it's great that we actually have more vaccine candidates because we're going to need them. And I think, as was alluded to earlier, this is a living guidance document, and a lot of really the majority of the recommendations are sort of based on what was the vaccines that actually had emergency use authorization at the time. And so this is actually going to be a very intense focus of discussion, I think, for the next round of discussions. I will say that as an infectious disease specialist, these adenoviral vector vaccines can be classed into two categories: replication competent or replication deficient. And the ones that are emerging late in phase 3 trials, such as the Janssen J&J vaccine and the AstraZeneca, these are replication deficient. Now, the way they've been engineered is they've actually had deletion of key parts of the adenovirus, E3 and E1 cassette, that actually render it unable so that it cannot replicate. And from my standpoint, I do not consider this to be a live vaccine. So it's as if you've kind of gutted out the inside portion of the adenoviral vector and then put in the DNA coding for spike protein. And so that these vectors, what they are just delivery vehicles to actually get the DNA to actually produce the spike protein and generate the immune response. And so I think that has been a source of confusion. And I think it's very important that each viral vector vaccine be considered on its own, but really the ones that are coming are replication deficient, and I don't consider them to be live vaccines. They're just a vehicle to get the DNA to produce the spike protein.
Okay. Dr. Calabrese, I was going to ask you one of the questions that came up, which I think you addressed to some degree, but to maybe touch on a little bit more, is sort of this patient, that we all deal with, unfortunately, with bad vasculitis who needs rituximab and needs it yesterday, and maybe before a vaccine is available. What advice do you give to such a patient and maybe a provider who's struggling, through that? What do? Do you dose the vaccine? Do you try to delay it? I guess what are your thoughts on that?
Yeah, that is the challenge, and that's a perfect example of how our guidance will serve as a springboard for case by case, shared and informed decision making on what is best for that patient at that moment in time. And in particular, we use rituximab a lot in vasculitis patients who have threatened end organs, and if they're having acute or new onset or relapse disease, those are important treatment decisions. And I think the decision comes down to what's best for the patient. In the specific scenario, is it better to administer the vaccine and assume you'll have a very blunted response, perhaps some response, we're not exactly sure, versus no response at all. A perfect example if this is last week, I admitted a patient with rapidly progressive MDA5 myositis who was in between her COVID vaccines and got some pushback about giving rituximab in-house because she needed her COVID vaccine. She was very sick and had threatened end organs, and that was a situation where that patient got rituximab, and whatever the immune response for her vaccine given afterwards is what it will be. And I think that's a case-by-case basis and severity of an extent of the disease and acuity of the end organ involvement. So a risk-benefit discussion with the patient. That's a really big challenge, though, and there's no right answer to that question, I don't think.
All right, great. Thanks, Cassie. That was very informative and helpful, though. So I'd like to go back to some of the questions that were presented by the town hall registrants. So going back to you, Dr. Arasaratnam, you're a transplant ID specialist, and many of your patients are on high-dose corticosteroids. So we've had a lot of people ask us to address the extent to which various doses of systemic or even intra-articular glucocorticoids might impair the vaccine response, and what you might say about timing considerations related to vaccination.
Yes, thank you for the question. And there may be others can weigh in about the intra-articular steroids, but essentially, we think of, you'll notice in the document that there wasn't a consensus regarding the vaccination when it comes to those above a daily dose of prednisone 20 milligrams daily. And I think it just gets to the nuances and the challenges of this, because certainly from an immune standpoint, if you look at documents such as the Infectious Disease Society of America vaccination in immunocompromised in 2013, they used prednisone 20 milligrams to denote a high level immunosuppression, and that's certainly been perpetuated in several guidance documents. A lot of that sometimes relates to the risk of live vaccines, which is not really the issue here. And what we're really looking here is actually the impairment of immunogenicity and how that translates into efficacy. For the Moderna phase 3 trial, those on prednisone 20 milligrams daily or more were excluded. And so we know from other data looking at flu and pneumococcal responses that doses at this range or below are probably unlikely to have significant impairment on antibody titers. I think we can all agree that in the middle of a high dose pulsing of steroids, administering a COVID vaccine in that setting is unlikely to generate the immune response wanted. And similarly, those on chronic stable low doses of prednisone, we would be very encouraged to vaccinate. But the problem is that there's a gray area in between. I would say there's nothing magical about the 20 milligrams, and I would really consider about the kinetics of steroids in your patients, as which way they're heading. What are the other concomitant medications that might impair immune responses, and also the individual risk of COVID. And I would really emphasize that we should be looking to vaccinate where we can, and this is not really intended to set up a barrier. Now, when it comes to intra-articular steroids, I think there has been some guidance come out from American Academy of Spine suggesting about waiting 2 weeks after that injection to get a COVID-19 vaccine. I would say that the personal opinions of the levels of systemic concentrations from an intra-articular injection, I do not feel would be significantly long enough to sort of impair immune responses. The one caveat might be is that we know that there's reactogenicity surrounding this vaccine, including joint pains as one of those things. And so to avoid conflicting presentations of disease versus vaccine reactogenicity, there may be a need to space. But I would really emphasize trying to vaccinate as soon as possible and not getting too hung up on specific numbers. And that is really why consensus couldn't be reached for above 20 milligrams of prednisone daily. And Dr. Karp, if others have more to comment on this, I'd be happy to hear their thoughts too.
Okay. Thank you.
I completely agree with you. I would just throw in, as a rheumatologist and infectious disease physician, I don't often think of systemic or intra-articular glucocorticoids as being systemically immunosuppressing most of the time, and I think most of us would agree. But a lot of orthopedic surgeons at my institution asked me about this today and made me think about it a little bit, and they seem to be hung up on this a bit and are into timing intra-articular injections around COVID vaccines specifically, which I don't think there are any data on intra-articular injections impacting any vaccine responses. And those Academy of Spine Medicine recommendations do exist for COVID vaccine, but I think there are no data for that, and something to consider in certain scenarios.
Great, thank you. We have the next slide, please. So, Dr. Bass, this is a question that comes up very frequently. In fact, I had a patient ask me this today. So, can you address the potential role of measuring antibody titers in some patients to assess immunity or lack thereof from vaccination?
Yeah, this is a great question. It's the question that we all have and we all want answered. The answer is that someday yes, but right now no. And the reason for that is that many, if not most, serologic tests that are commercially available for COVID infection measure antibodies to the nucleocapsid protein. And the reason for that is that antibodies in patients who develop COVID, antibodies to the nucleocapsid antigen develop earlier. And so those assays are more sensitive. That serologic test is more sensitive than tests for antibodies against the spike protein. But the vaccines are directed against the spike protein. So if you do a nucleocapsid serology, you're going to get a negative result, and that doesn't tell you anything. But even if you have an assay that is measuring antibodies against the spike protein, the problem is that we don't yet have any correlation between levels of antibodies by those assays and seroprotection, protection from infection. And we need those before we can tell a patient your level of X antibodies is enough to protect you, but somebody else's level of Y is not. This is clearly needed, particularly for our patients. This whole discussion about rituximab, if we had guidance, antibody responses that we could measure, we would know whether patients need boosters, and we could make data-driven decisions about when it is appropriate to vaccinate after rituximab dosing. So for the future, I'm hopeful that this will be available, in the next 6 to 12 months, but right now, unfortunately not.
Great. I know there are lots of labs around the country, including some of the people here at our institution who are looking at this very question. And as you say, hopefully, not 6 months from now, but maybe even 2 or 3 months from now, we'll have some great answers for people on that.
Fantastic.
So I'd like Dr. Seddon to field this next one, which actually has a number of parts, but refers to vaccinating younger patients. Can you speak to the use of the vaccine in patients who are 16 to 18, or the off-label use in younger patients, and any information about vaccine trials in pediatric patients?
Yeah, all great questions. Thank you. So the first thing to note is that our committee recommended full support for following the emergency use authorization. So for Pfizer, that's going to be patients ages 16 and up, who those are the ages that were in the trial, and for Moderna, that's 18 and up. Now, the question of off-label use for younger children is honestly probably moot right now. Children are not in any of the priority groups being offered the vaccines per CDC guidelines or state guidelines. But the good news is that there is more information coming down the pike. So Pfizer has already fully enrolled more than 2,000 patients in its study of patients ages 12 to 15. So the hope is that in a matter of months, we'll have information on that age group, and that the EUA guidelines can be modified to include vaccination down to age 12. And the plan is after that to have another trial of patients even younger. So I think the hope is that by fall of next year, school-aged patients and up will hopefully have data on their vaccine. And then with regards to the multisystem inflammatory syndrome of children, so there haven't been any noted cases. Bear in mind, of course, there haven't really been many vaccinated children, but we haven't seen a corollary in adults either. We haven't seen far downstream major multisystem inflammatory serious adverse effects from any of the vaccines in any of the trials.
Great, great. And the last submitted question, next slide. So for you, Dr. Calabrese, we've had a number of questions that related to COVID-19 therapy and vaccination. So can you address how long to wait after remdesivir or convalescent plasma before getting COVID-19 or vaccine, and what to do if a patient gets the first dose, then develops COVID-19 and treated with the above medications? What do you do with the second dose? Seems like a strange situation, but it's likely to occur.
Yeah, for sure. Thank you. These are good questions. Talking about treatment for COVID-19. So first, remdesivir, which is authorized for treatment of certain hospitalized patients with COVID-19 on some degree of supplemental oxygen. There are no current recommendations to delay COVID vaccine at all in someone who has received remdesivir. The convalescent plasma story is a bit different. This is authorized for treatment of outpatient COVID-19 infection. And while there are no data on COVID vaccine efficacy in patients who've received convalescent plasma as treatment, based on the estimated half-life of the convalescent plasma and the low likelihood of reinfection within 90 days after having COVID infection, it's recommended by CDC to defer for 90 days after infection. This is theoretical. Until we have more data, could convalescent plasma interfere with vaccine efficacy if given within that window? The question of what to do, and this applies to if someone is naive to their COVID vaccine series, or if this is administered between vaccine 1 and 2.
Great, great. Okay, so Dr. Michaels, we have a few minutes for an additional question, or I think that may have come up.
Sure. There was, I think, one really interesting question about, we've heard a lot in the lay press about people with reactions post-vaccination, and I don't know who would like to try to tackle this really easy question, but what do we know about adverse effects in the rheumatic disease population? And is it possible that some of that may be related to the vaccine carrier, so the lipid layer used, rather than maybe the mRNA, or perhaps polyethylene glycol that's found on the 2 mRNA vaccines? What do we know about these other components? Who'd like to try tackling that one? Ruben, do you want to take a shot?
Yeah, I can definitely take a shot. I think I think teasing out these adverse reactions are challenging. I think there has certainly been, for true severe anaphylactic reactions, protocols developed in place for those who may have an allergy to polyethylene glycol. It's commonly found in gels, bowel preparation, and there's a derivative of that which is in part of some of the other future coming vaccines of polysorbate. And so that's been moved in the allergy community to actually try and sort of tease that out separately. Beyond that, I think to sort of look at the different vehicle carriers and the lipid carriers and say that it's responsible for certain symptoms. We just don't have the data. All to say is that, you know, there is a set pattern that's seen in these phase 3 trials of reactogenicity. And to me, it's not surprising that when you have 2 mRNA vaccines that are over 90% effective, that we're going to see some, you know, systemic symptoms that are noticeable. Malaise, joint pains, fevers, and sometimes the overlap with those reactogenicity symptoms with underlying autoimmune conditions is challenging to tease out. And so all I would say is the best thing we can do as providers is to use the reporting mechanisms that are in place as much as possible to give the true denominator of this data of what's going on. Make sure that these are areas of research priorities and studies, and that's part of the document too, of actually highlighting certain areas of research. And I think that's all I can probably speak to with regards to that one.
Okay. There's been a couple questions, and I think this speaks to how rapidly things evolve. You know, you put together a guidance document like this, and the evidence is, you know, emerging before our eyes. There's been some reports of ITP or immune-related thrombocytopenia post-vaccination. Dr. Anthony, I see you shaking your head, so maybe you've heard the same reports. You know, would you care to comment on, you know, and I know this is outside the realm of the guidance document for sure, but any comments on sort of your observations or thoughts? I know that the reports have been pretty small in number, given the denominator of people who have had the vaccine at this point, but I'd be interested to hear your thoughts on that.
Yeah, I've not reviewed any of the information regarding those cases, and short of the understanding that these vaccines appear to be quite unique in their ability to induce an immune response, elicit a very potent immune response in a short time, do have us with the understanding that there's probably more to learn about mechanism by which they work, and potential side effects. I think, as has just been discussed, though, this is all counterbalanced by how many individuals have truly received the vaccine, and discussions that we had as a group included discussions about how many of those people that were in clinical trials had unknown autoimmune disease or predisposition, and yet we did not observe autoimmune flares in those people with occult or subclinical autoimmunity. So I think we have a lot to learn is my best answer.
Thank you. I think that's goes with for all of us. There's been a few questions as well on the CDC has guidance about pre-medication treatment, and so there's been some questions around NSAIDs, and you know, do our recommendations touch on that? Would there be comments on NSAID use in general around the time of vaccination? Jeff, maybe I'll give that one to you and just your thoughts. And there's also kind of a similar question around apremilast and whether, you know, we should be thinking about how we treat that agent in the context of vaccination.
Yeah, I think that in general, there's really nothing that the task force said that would in any way contravene the CDC guidance. In some cases, it refined them or called out some specific examples or applications. But I think that, you know, unless you have data that would say we think that the CDC is wrong, I think in general that the task force is going to more or less align with what the CDC has recommended, absent evidence to the contrary. The full manuscript will include that and some additional details where frankly we just point back to, hey, the CDC said this, and you know, Rebecca and others even on this call sort of pointed out that the task force mostly has just echoed what the CDC has said about use in pregnancy and lactating women, and I think we continue to do so absent evidence to the contrary. And likewise for apremilast, you know, I think most people and evidence suggests that this is not particularly immunosuppressive, and something that we need to be very concerned about, attenuating vaccine response. So I wouldn't anticipate that there would be much enthusiasm to modify either the use of that drug or its timing or the vaccine timing in relation to that treatment for people that are on that therapy. But certainly the full manuscript can address that in a more complete way.
Thanks, Jeff, and thanks for all your work and leadership on this. I was going to take the prerogative, hopefully David doesn't get mad at me, but maybe throw a question at our president. And there's been some comments on here about, struggling to get our patients vaccine access, and in terms of different CDC classification groups, and advocating for rheumatology patients. And I think our members on tonight would love to hear a little bit more about the ACR and how we're advocating for our patients.
Well, thank you, Ted. And I would just say that probably the greatest advocacy for our patients is the fact that we produced this guidance document, because right in the very beginning of the document, it says that patients with chronic autoimmune and inflammatory diseases need to be prioritized for the vaccine. And so, unfortunately, vaccine priority is a state issue. And that means that you have 50 states that you need to work with, and the ACR has a large network that interacts with state rheumatology associations, and takes its advocacy down to the state level. So if there are particular states that are being recalcitrant in this area, we are happy to work with the state organizations and provide as much information as we can, but most of it is going to come from this document. I was happy, Texas may not have a great power grid. We're sitting here, we're hoping we don't get blacked out in Dallas, but tonight, but we have been able to get the vaccine to almost all of our patients in the rheumatology clinics here, because the state has chosen to allow them to be prioritized as 1B here, not as 1C. So we're very lucky, but that just goes to prove that this is a patchwork thing, and we need to work together on this. Can I have the next slide, please? So I'd like to thank the rheumatology community for your questions and engagement this evening, and special thanks to our panelists for their insights tonight. Our goal has been to assist the rheumatology community with clinical recommendations during this pandemic, through the work of multiple task forces that have developed adult rheumatology, pediatric rheumatology, MISC clinical guidance, practice and advocacy resources, and now vaccine clinical guidance. All of these resources can be accessed via the ACR website at www.rheumatology.org. Next, please. And thank you again to everyone who participated in this town hall. And if you have additional questions, please email the ACR at covid@rheumatology.org. This recording will be available on our website at www.rheumatology.org shortly. And it's the ACR's promise that we are here for you so that you can be there for your patients. Thank you and good night.